I'd like to welcome everybody to our first Health and Sciences Colloquium for the 2012-2013 year. I'm Kathy Finan. I'm one of the bio faculty here, and I would like to welcome and introduce you to Dr. Joseph Orgel from the Illinois Institute of Technology. He is Associate Professor of Biology and Biomedical Engineering. He received his bachelor's degree and his PhD from the University of Stirling in the UK. His research focus has been on the molecular structures and the function of extracellular matrix. This is the material in living tissues that's found outside of cells. So it has a lot of different functions, and it's like so many other things in biology, its function and its structure are intertwined very intimately. We're going to be learning something about that today. Dr. Orgel is also the chair of the American Crystallographic Association. He has many, many publications to his credit. And in 2007, he was the recipient of the National Science Foundation Career Award. This is an award that's given to junior faculty who are just starting out in their careers as academics, and it's given for excellence in both research and teaching, and the ability to integrate the two. So we're real fortunate that Dr. Orgel is joining us here, and you take it away.
Thank you so much. And with a great introduction like that, with expectations high, there's only one way I can go, which is down, as soon as I open my mouth. So if you'd lock the doors, that would be great. We'll be here for the rest of the evening. Before I really bore you to death with the world's second most boring protein, which is, yeah, I'm really trying to lower the expectations again now. Can you tell me what you know about the world's second most boring protein? Can you tell me what it is? Give that man a star. What, say again?
It's used in a lot of plastic surgery.
Used a lot in plastic surgery. Why would that be?
It's the connecting tissue and muscle fibers.
Right, it's the connecting material, you would say.
Sorry.
No, by all means. Tissue is a conglomerate organization. Tissue can just be cells stuck together. Commonly, tissues are extracellular matrix material with cells. The two of those make up a tissue as such. Excellent. What can you tell me about collagen structure? It's a long chain. Somebody higher up. Yes, hello back there. What can you tell me about collagen structure? Yeah, well, we'll go with you, sir. It's an alpha helix. I will say helix. It's helical. What else can you tell me? This gets me kind of up to speed. I don't really have to say very much. Give the lecture for me. All right, so you know, so far I'm going to take a synopsis that as a group, you know that collagen is important. And I will say that its importance is underpinned by the fact that it is the most abundant protein in the animal kingdom. It underlines, it is the basis for the connective tissues. And connective tissues are structural support tissues. Skin, bones, tendons, eyeballs, ligaments, teeth, cartilage, and in fact the material that gives structure to organs such as the heart. You see, I'm telling the truth because I have pictures. If you decellularize the heart, that is, if you remove all of the muscle cells, that's removing all of the muscle from the heart, what you're left with is a horrible mess, yes. But what you're left with is something that still looks an awful lot like a heart. You can then populate that, you can use it as a scaffold, and populate that with mesenchymal stem cells, adult stem cells, and you can use those to differentiate themselves into heart muscle cells and become a brand new heart or re-engineered heart for the genetic host who donated those mesenchymal stem cells in the first place. But apart from being an organizational entity, apart from being something as a scaffold upon which you can build attachment sites and organization sites, and in fact the organization of tissues, it's also an information molecule. And this is something that most people don't think about. You think the paradigm for proteins is that they are either fodder, i.e. food, or they are enzymes that act upon other parts of metabolism, right? It's a leading question, but in this case, I would say right is a good answer. Structural proteins are not just there for structure, but if you really think about it, structure implies information and organization. Now, how it works within the body is a cell doesn't know what it's supposed to be to start with until it reads its environment. It reads its environment and it says, from the context I'm in, I'm supposed to be an osteoblast. Let me look through my instruction manual about how to be an osteoblast. So it goes to the nucleus and it finds the genetic information that tells it how to become that, and it becomes that. Cells are also responsible for making the environment in which they situate. I hate saying chicken and egg, but it's that kind of idea. So somehow cells both determine what they become because they determine what the environment is, but they also become what they're supposed to be because the environment determines that. And a big part of what the environment is a treasure chest full of pirate booty ready to be taken by ye scurvy dogs. No. A cell would look at a collagen fibril, which is the entity of which collagen appears. And I'm going to spend quite a while telling you what a collagen fibril is, because it's kind of important. Looks at a collagen fibril and goes, well, that would be yummy if there was something in there I could get to. And then something happens, and they find a collagen fibril is full of chocolate gold. No, it finds that it's full of things that cells want to take on and attach to. So the chocolate inside this treasure chest is representative of amino acid sequences that a cell regards as instructions to tell it what to be and where to attach on the scaffold for the tissue. So here's part of the motivation, and I'm not going to show any more pictures that are more distressing than these, as far as I remember. These are various pictures of rickets. This is a nutritional deficiency that results in a weakened skeleton, and these are for juveniles, and therefore even more distressing than usual, but anyway. These are sections of transmission electron micrographs, so they're microscopic pictures of mammalian joints. This is the healthy joint, and anyone with arthritis will recognize what it feels like to have joints that have these kind of unsmooth edges. That's osteoarthritis, or joint damage in general. Sports injuries can end up making your knees and ankles look like they have edges like that. And this is rheumatoid arthritis, which is an immunological disease which leads to a breakdown of cartilage, much unto like this. Now, interestingly, in our research, we found that there are cartilage-like structures in the human heart, which goes against perceived wisdom, which just goes to show you how wise perceived wisdom really is. The cartilage-like structures, I'm not talking about knee cartilage. I'm not talking about nose and ear cartilage kind of things. I mean on the molecular level. Organizations of collagen that are reminiscent of cartilage. That being said, we found there's something in the septum, in the heart wall, between the two sides, between the left and the right side, which makes sense in terms of the compressive forces that are being applied there, but also in the valves as well. And we found what we believe to be a link between rheumatic fever, rheumatoid arthritis, I'm not saying those two are linked, but those two things on one side, and ventricular failure on the other. And this is a current part of our investigative process at the moment. So this is pretty much saying what I've already told you, except with a pretty picture on the background. This is an atomic force micrograph of type 2 collagen. This is the primary collagen that's found in cartilage. So the collagens, there are 29 of them, maybe 30 of them that have been classified so far, and a subsection of that family of collagens are fibrilla. They make fibrils, these things. This is a type 1 collagen fibril, and you may notice that there is something of a repeating pattern in this collagen fibril. And it's obvious in this instance because this sample has been treated with heavy metal stain. It's a negative stain. And we haven't talked about this yet, but there is a crystallographic, there's a crystalline repeating property to fibrillar collagens. And that is each of these dark and light bands, we'll call two of these together a D period, so dark and white, dark and white, dark and white, dark and white. That's free D bands. In the dark band, in that space, there are repeating entities of 4 collagen molecules, and in the light band, there's repeating entities of 5 collagen molecules. There's 1 collagen molecule less in the dark band. We call that the gap region, because there's 1 less, there's a gap. Because that's actually a gap, when you put heavy metal upon the sample, more metal gets into the gap region than gets into the light region. Therefore, in this technique, it looks dark. So this is something that's quite common when you come to look through the literature. It's quite common to see pictures like this, and now I've just very naughtily taught you something sneakily. The distance between the start of this dark band and the end of this light band in a native natural collagen fibril is 67 nanometers. And that number will matter in a moment. But that rope-like structure is what most people think of when they think about what collagen is, what collagen fibrils are. Very large, something that the scale here is suggesting that that's 400 to 500 nanometers in diameter. Because of the work that I do, I don't think actually that's a collagen fibril. I think that's a fibrillar bundle. But I'll explain what that means in a moment. Now, here's another way in which people think about collagen. So you've told me that it's a helix. Actually, a single collagen molecule is a triple helix. And the triple helix is because there are 3 polypeptide chains in each collagen molecule. Each of those polypeptide chains have a left-handed polyproline 2 helix, a curly thing going this way. And each of those things going this way wrap around each other with a rightward sense. Most cell biologists think of collagen in these terms, amino acid sequences. If I was to draw the collagen molecules sequence, the whole thing out here, and I really don't expect anyone to be able to read this, and that's part of the point. The end of the sequence would be at the car park A, or the premium parking, probably to scale. And then this would be the other side of the building, down to whatever the main road is out front of the building. There's 1,054 amino acids in the alpha-1 chains of the polypeptide. There are 3,150 amino acids in each collagen molecule. It's enormous in molecular terms. But the minimum collagen entity that a cell cares about is not 1 collagen molecule. It's about 14 billion of these put together to make a collagen fibril. And what my lab is known for is for determining the structure of the collagen fibril to atomic resolution. And that's been kind of useful for making sense of this nonsense. And this really is nonsense, because what we're looking at is short sections of amino acid sequences that we're then trying to use to understand how cell biology works. Now, something you have to understand is that cell biology, as with a lot of science, a lot of discovery science, is rather like performing discovery car mechanics with artillery fire. So you blow the thing to pieces, and then you pick up these charred, burning embers from the car, and you're like, I think this might have been a fragment of something. We'll call it a carburetor. And then you argue for 40 years about what a carburetor is supposed to look like. So what we try and do is study the structure of the carburetor in situ, in the car itself. Yes, so take that back to what I'm really talking about, of course, is the structure of the collagen fibril and the tissues in their original context. And that involves a variety of techniques. But in addition to the techniques that we've developed to look at these things, you also need to be able to interpret the great wealth of data that's out there. So what we do is take these little amino acid sequences and put them back into their context. So we work out what the structure of the collagen fibril is, and then we take these little amino acid sequences that we know mean something about cellular interaction, but we put them into the models of the overall structure to see what they mean. Now this is important because this is the cell hotspot. We call it the cell interaction domain. That's a part of the collagen molecule that cells really, 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 really like. I think now we've called it the interactosome. I get lost with what my collaborators are up to with that. Sooner or later we'll come down with something. I like hotspot, so let's go with cell hotspot. A couple of things we're going to come back to later is this is an integrin molecule. In this cartoon, we're representing an integrin molecule. Integrin is what cells chiefly use to latch on to collagen with. And those integrin molecules span several domains. They span the inside of the cell. They attach to the cytoskeleton on the inside of the cell. They have a transmembrane domain, so they cross the plasma membrane. And then they have an extracellular domain represented by these two balls on the end of the stick, which they latch on to this hotspot, and maybe a couple of other spots as well, and the collagen molecule. And cells attach themselves to this. Also, they can detach and then reattach. They can walk along a collagen fibril. And in fact, they do. For fibroblasts and for tissue synthesizing cells, such as fibroblasts, osteoblasts, chondrocytes, and so on, those are skin, tendon, cartilage, and bone synthesizing cells. They like to stretch the tissue. The tissue works better if it's synthesized under tension, and so do the cells. And then there is these little wrapper things. So you see these curly long chains here. Those are long sugar chains with a little protein attached to them. Those are called proteoglycans, and you can think of them like compression straps. So if you have a large truckload of tree logs on the back of a truck, then you use these large ropes and compression straps around them to stop them rolling down the hill and killing people, rather to stay on the truck. And that's kind of the equivalent thing that proteoglycans do for collagen fibril. So imagine each of these little entities here as a collagen fibril. These carbohydrate chains are wrapping around multiples of them to make a much larger bundle, a fibril bundle. But to say that this particular diagram is accurate is like saying this is a good representation of me. It's got a beard, and that's exactly where I'd go to. But you know, depending on the quality of the technique used to capture me in this image, if I was a critical reviewer, I'd say, you know, I don't think that your technique had the resolution to tell that was a beard versus an artifact. Believe me, I've had reviewers like that. It's a beard, people. Anyway, this does some useful things. It gives you the basic pattern of a Joseph Orgel. There are some problems, some details missing, but it gives you some real details missing, but it gives you an idea of what an Orgel should look like. Oh, you went somewhere I wasn't going, but maybe later. So anyway, what I want to do is take this idea of the cartoon and to do something 21st century, which is not iPhone related, but instead to take atomic coordinates that we have developed from experiments working on the native structure and all of the information that we can find, no matter how horrendously complex it is, and it is, believe me, horrendously complex, and move from the tune to actually something that's quantifiable and we can use to make a map of the animal body. And we find in this odyssey that as we travel along in this voyage of discovery, we find enterprises to go in search of. And I'm not thinking about sci-fi TV series, but I'm talking about clinical things that we can do. Making bioscaffolds to treat disease, finding diagnostic things that we can do, and yes, collaborating with paleontologists to do dinosaur research, which is why a bunch of you are here. Anyway, I will go through a very simplified bit of, what did you think I said? Bit of crystallography. People are going to be playing that on YouTube for a while to determine what he really said. I hold my bit. Anyway, these are some of the samples that we're looking at. They are lampreys, relatives of hagfish and eels. They're notochord. A notochord is kind of equivalent to vertebra even their relatives in the same family actually have vertebra. However, the notochord surrounds their spinal cord, and it's a protective tissue. The notochord in these creatures is very similar in certain critical respects to the cartilage found in animals and humans, which makes it a good model for investigative studies such as me. And of course, for a variety of reasons, which I have no idea why, but anyway, for a variety of reasons, the diffraction pattern is missing from this slide, and there'll be some other artifacts in this slide later on. But anyway, this is a pig's eye, which of course you could tell, and this little white spot here is a bit of the retina near the intrusion of the optic nerve into the retina inside that eyeball. I did say there wouldn't be any more horrible pictures. You look horrified, my dear. You've been horrified for a while. Anyway, what we did is at about 3 a.m. One morning back in 2007, I shot X-rays at this little thing at the advanced photon source, and I got this diffraction pattern, which I was so pleased with, I'm still showing it 5 years later. This line here is a function of the spacing between amino acids in the collagen inside the optic nerve inside the retina of this pig. I'm sure the pig is delighted to know what its eye was donated for. Anyway, a lot of the research that we do is actually pretty simple. I collaborate with a number of investigators looking at various tissues, biomaterials, and even non-biological materials. And I'm able to say, hey, your molecules are spaced 3 angstroms apart. Have a nice day. That's pretty nice. But when I want to do something really complicated, I go to the workhorse, the rat, the white rose carrying rat, and I say, please, Mr. Rat, give me a tendon from your tail. And he says, of course, for you. And then I shine an X-ray through the, I shine X-rays through the, this is when you're trying to be funny, everything goes to pot. Shine X-rays through whole bits of tissue, such as tendons, and get these very aesthetically pleasing diffraction patterns. Now, there is vastly more detail in this diffraction pattern than there is in this one, because the molecules in the rat tail tendon are exquisitely well ordered. And in fact, I found that this is a theme in a lot of connective tissues, that when you treat the tissues very carefully, very gently, and keep them as in their native state that they were in the tissue, other than the fact it's no longer alive, what you find is that the tissue has crystallinity to it. What does crystallinity mean to you? I've said this about 7 or 8 times during this talk. What does crystallinity mean? It's a defined structure. It's a solid, yes? Lots of symmetry. Do you all have a general sense of what crystalline is? So you may be able to divide a crystalline substance into subunits, where any subunit would define the whole. And the subunit would be the unit cell. So you can find a subunit of your crystalline solid, and from that one little bit, you could define the structure of the whole, because it's a repeating crystalline structure. The crystallinity of rat tail tendon, we have data to below the amino acid residue level, which is about 2.86 angstroms resolution, which most single crystal crystallographers would be absolutely ecstatic to get. Oh, by the way, I'm not the chairman of the Crystallographic Society. I'm the chairman of the Fiber Diffraction Significant Interest Group. It's a bit of a difference. I'm still important, but not that important. Right. So this is a little portion of the electron density map. An electron density map, as it implies, is showing basically where the electron clouds generally are surrounding the atoms within the molecules that they comprise. And so the blue wireframe here is showing the outlines of tubes. Those tubes are collagen molecules. And in the middle of these tubes, I've built in some atomic models. There's a red model and a yellow model there. And if this was a much more aggressive, detailed talk, I would spend a while telling you about those models, but we're not doing that to you because this is simplified crystallography. However, the collagen molecules are packed together in such an accurately repeating fashion that I'm able to define where the amino acid residues are and some of the atoms within them based on the X-ray interference between the packing of the collagen molecules. And I said that sentence for maybe 2 people in this room. And if you found that interesting, ask me questions later. Anyway, you remember the D period that I was talking about some moments ago. And I talked about a dark band and a light band. Well, this is the molecular model for a D period now. So we've gone from a squizzy dark and white image that we had a few minutes ago to, hey, look, here's the molecules. And in the overlap zone, which is about here, bear in mind that yellow wraps around from side to side. So it's not 2 yellow molecules, it's 1 yellow molecule. That's 1/2 of it split on this side. And then on the opposite face, there's the other side. Because this is a unit cell from the collagen fibril. And there are 4 collagen molecules in the gap region. Now, because we've got this, suddenly we're hugely empowered because we've got a unit cell of how collagen molecules are organized in nature. And this data was obtained from whole tissue, from a tendon. So suddenly we've got the archetype for all fibrillar collagens because we've got type 1 collagen structure. And we've certainly got an accurate map for type 1 collagen structure regardless of what tissue you're in. Because I haven't told you this yet, most of your tissues, most of your connective tissues, ligaments and tendons, for instance, are made from essentially the same ingredients. They differ because the recipe is different. Does that mean anything to you? You can immediately tell who cooks on a regular basis. So you may have the same things in there, but what you do with them can make a big difference. It's like eggs. Scramble them and omelette. I mean, what's the difference between a scramble and an omelette? Not that much, except I like omelettes and I don't. Anyway, so the top part of this this whole thing is supposed to represent a part of the X-ray diffraction pattern. And just so you know, this top part here is representing data that I collected from the tendons, and the bottom part is a transform, back Fourier transform of this to confirm that we were getting somewhat close to the right structure. That isn't how I solved the structure. It was just a final check. So the triple helix that we were talking about, this is here. And the fact that collagen, fibrillar collagen is comprised of triple helices has been known for about 50 years. The rest of this information here, anything that's marked with a red asterisk, is essentially what my group has done in the last 10 years. And the other things that we worked out. Because there was a considerable impasse in working out what the structure of collagen is, beyond, hey, look, it's a triple helix. These little bits at the end here, they're called non-helical telepeptides. They're essential for collagen structure. They're the points where cross-links are made between one collagen molecule and another one. And when you have those cross-links, you're able to make long fibers. So remember I told you that each of these polypeptide chains is one polypeptide chain. It adopts a left-handed helix. 3 of these wind together in the opposite direction to make a right-handed superhelix. Superhelix, triple helix in this context is the same. And then 5 of these superhelices wrap around each other, again in the rightwards direction, to make a super helix. And that's just ridiculous, so just call it a microfibril. Because super duper, no, that doesn't make sense. Let's not go there. So I've labeled here the 67 nanometer repeat for you, so you've got some kind of idea to be able to follow things through. And you notice it's got this kind of squiggly back and forth, this wiggly wobbly kind of, was it wibbly wobbly timey wimey thing? This back and forth pleated structure, which is reminiscent of the fibril. So this is a little section of a collagen fibril again. You see the inny and outy bits? It's reminiscent of the microfibril. It's part of the reason why you can think of this as a microfibril. So some things I want you to keep track of. C-terminal telepeptide, this guy here. C-terminal telepeptide is right on this most bumpy bit. Let's call it the nose. Right on the nose here. That will be important for a couple of slides from now. I did all of this so I could get to this, which was to work out how these microfibrils are arranged in a fibril. Because all of that exciting amino acid chemistry, all of that chocolatey goodness inside the treasure chest Not all of that is available on the outside of mature fibril. Cells are not allowed to get to the chocolatey goodness until the chocolatey goodness is ready to be got at. So to be more direct, those cell attachment sites are not available for cellular attachment until the tissue is ready for cells to attach to them. Because if it happens willy-nilly, that's bad. It's called disease. If it happens in a strategic way, that's normal growth, development, or repair. Now, so that's why this is important, the way in which the collagen molecules are organized in the fibril. So what we've done is now we know where the molecules are, where the amino acids are. Now we can work out what sequences are available and what's not. So this is a surface rendering of the collagen fibril. This is not what cells would see. This is what we see as if we're pretending to be a cell. And I'm showing you a little bit of the structure around the C-terminal telepeptide here. So this is this. That green thing there is the cell attachment hotspot. It's not very available for cells to attach to in the mature fibril. This yellow thing here is the strategic site for cutting collagen. Because something I haven't told you is that from this work over the last 10 years, I've come to the conclusion that outside of the cell, there's the same mode of operating as there is inside the cell. Proteins become active by proteolysis, generally speaking. There's usually a change to a protein to take it from an inactive state to an active state. And quite often, proteolysis is the way in which that happens. That means cutting it, removing a bit. And so I think that collagen in the collagen fibril becomes available for cellular attachment through the same paradigm, proteolysis. There's only one place that happens in the whole of a collagen molecule, unless everything has gone to hell in a handbasket, and you have stomach digestive enzymes running around the rest of the body. There's only one enzyme that does that in the rest of the body, the collagenases, and they do that here, at this yellow site, which is also protected by the C-terminal telepeptide. All of this will start to make sense quite quickly now, because I've spent half an hour giving you a crash course in collagen structure, so I can give you the punchline. Oh, yeah, it's not this. Please don't get overexcited or underwhelmed. But as I was saying with the artillery fire car mechanics, so far it should be apparent that working out how a tissue is put together on the molecular level is a lot more complicated than what's the sequence of amino acids. Glycine, arginine, cysteine. Does the sequence of amino acids mean? Who does it not mean anything to? Good for you, good for you. So amino acids are the building blocks of proteins. And the names that you call them relates to the chemistry that each one has. So when you tell somebody who knows about amino acids what amino acid you have following another one, that gives you an idea of what the properties of that protein sequence might be. Better? All right. Better, yeah, there's no way we can. It's a sticky plaster on a bleeding hemorrhage. Yeah, it's not only going to go so far. Or a Band-Aid, I should say. So right now we know which parts of the collagen molecule are buried and inaccessible until something happens. Either damage, which the tissue knows it needs to repair because it says, well, hey, there's stuff here that we don't normally see. Quick, deal with it. Or, oh, there should be stuff here about here, let's dig until we find it. Oh, there it is. That's what I can attach to. And the interaction of the cells requires other molecules. How those molecules interact with collagen, again, is a lot more complicated than the amino acid sequence. It's the shape. Does the key fit to the lock? It's the classic enzyme catalysis paradigm, for want of another phrase. But how things fit together is basically how molecular biology works. So I'm trying to go from this cartoon to something that's a lot more quantifiable. So this is a little section of that surface of the collagen fibril that I was talking about before. So let's see where we can get to. Oh, that's much better. Certain key amino acids sequences that are important to cell function I've labeled on here now. This VWF is von Willebrand factor interaction site. Von Willebrand factor is a critical component in blood clotting. So von Willebrand factor binds here in the collagen fibril. But in an attacked collagen fibril, it's not very accessible, is it? Yes, it's okay to say yes. There's something sitting over the top of it. And in addition to that, the places where the proteoglycans sit, the proteoglycans are those compression straps that holds the logs together, remember that? Here and here, which also sit above the von Willebrand factor site as well. The collagen cleavage site is right here, marked in cyan, which is underneath this red site here. The red site is possibly the toughest part of the collagen molecule, the toughest part, which is that nose bit. You remember in the picture I said we'll call this the nose? The nose, the bit that sticks out most, is tough, is the toughest bit, as we might say. And then the cell interaction site, the hot spot here, is here as well, which is also basically inaccessible. But now we've got a way of doing molecular interaction studies, molecular dynamics even. So a few years ago, we published a study about how the protein bit of the proteoglycans, how they would attach to the collagen fibril. This player here, this is glycoprotein 6, is the major factor for platelet attachment. The initial attachment, blood platelets are essential for blood clotting as well. Blood platelets are cell fragments. They have a plasma membrane, but they don't have a cell nucleus. They are very important in enabling blood to clot when there's a wound. So I started putting these factors together and came up with a hypothesis for initial blood clotting, which is in the lumens, so in the holes of your blood vessels, when damage occurs, collagen's not normally visible. There's other things on top of it. So when some damage occurs to the lumen, collagen becomes available. Collagen is, here I am. But collagen does not have particularly strong binding sites for platelets to attach to. What it does have is this tough section here, right on the bit that juts out most from the collagen fibril. So in fact, this sequence here is the first thing that's most accessible to a GP6 molecule on top of a platelet. And these stringy things here are a rendition of the molecules on the outside of a collagen fibril. And this blue thing here is a molecular representation of glycoprotein 6. And you may notice that there is this good correlation between these docking sites of the two units of GP6 and the prospective binding site is this red thing, is marked in red here as well. They fit almost perfectly. Now the thing is that this structure and this structure weren't determined together. They were determined completely independently from each other. And no molecular dynamics or energy minimization was done. No trying to get them to fit together was done. We just took the two shapes, put them next to each other, they fit perfectly. So we looked at this, and then I remembered that reading that platelets also have certain collagenase enzymes in their plasma membrane as well. And a few years before this, we'd published a paper on how collagenase docks, right? So we worked on how collagenase docks as well. So we had a very good idea about how the digestion of collagen progresses as well. And so because we understood how the digestion works, we could see how the initial docking would lead to an activation sequence in the collagen fibril and make von Willebrand factor completely accessible for blood clotting to progress. So we've gone from having no idea how collagen actually works within the body to changing the field, changing the perspective within the collagen field, simply because we came across the emperor and his 3 blind wise guys trying to describe what an elephant looks like. We came along and went, oh, it's gray. And the big trunk is the trunk, and the little trunk is the tail. You want to stay away from that end. And suddenly, working out how a tissue is composed as we get these structures for these other extracellular matrix building blocks in relation to the collagen fibril has become quite simple and straightforward. So I'm going to speed through this a little bit and give you a punchline and talk about dinosaurs before I let you veracerate me with questions. So some of the things that we're doing now to try and work out where these extracellular matrix things are, these other molecules that help cells attach to collagen, and the other molecules that help bundle collagen fibrils together. These are atomic force micrographs. So this one is a native type 1 collagen fibril. And this is the same tissue sample, not the same fibrils, labeled with an anti-collagen antibody with a fairly large, I think it was a 30 nanometer gold particle conjugated to it. So we won't be able to see the antibody of this technique, but we sure as heck will be able to see the large spheroids, you see, attached to the outside of the fibril. And these are just various different ways of showing the same thing. So phase contrast, it's another phase contrast. Oh, actually, that's something I cooked up special anyway. I can't remember what it is, but that's an Orgel special. That's a phase contrast. And these are lots of blobby bits on the end here. Now, the funny thing is that I worked out in one of my most harebrained experiments ever, which actually turns out to be very reliable and easy to do, that we could take X-rays of these antibody-altered tissues and treat it as if we were adding electron density contrast to a crystalline entity that didn't otherwise change. So we take, say that this black line is the overlap of a D period, and it has a step into a gap region because there's less material in here. What I'm showing you is a 1D version, a one-dimensional version of a collagen fibril unit cell. Overlap, gap region. This is native. Say that I take my gold particle attached to the antibody, and I add electron density into one particular part of the D period. Suddenly I can do spectroscopy. This red line is the equivalent of this with the gold particle added and then this black subtracted. It's called a difference Fourier map. This bleb here, this peak, is showing me exactly where this is, except it's showing me at a resolution of 5 nanometers. And why does this matter at all? It's because I just found where the major histocompatibility site is for the fibril. That's where autoimmunity starts for collagen tissues. And it took about an hour. I should at least spend 2 minutes talking about this, since it's in the title. So we took our, understanding about collagen to a team of paleontologists who, several years before, had benefited from something that initially seemed to be heartbreaking, which was a discovery of in the Rockies of a Tyrannosaurus thigh bone that was fossilized but too large to move from the site. So this is a picture of the Museum of the Rockies, Tyrannosaurus thigh bone 1125. Or a section of it, and it was too big to be able to remove from the site without help with a helicopter, and in fact, the helicopter wasn't able to remove it from the site without the bone being chopped in half, which is something scandalous. You would never do that with a precious specimen. You've got to treat it gently and preserve it and do everything you can to preserve it. Do you remember how we discover things? Kind of blow it to bits. It's a very bad approach, but it's the best thing that we've got to start with until we learn a lot about it. Then when we learn a lot about it, then generally, if something's completely new, generally we take it apart to work out how it works. The first time I came across a grandfather clock, I took it apart when I was a little boy. And I learned lots about grandfather clocks. I wasn't able to put it back together, but never mind. What happened is that this bone was chopped in half, and the insides from the bone, which are not normally found for fossils, were analyzed by a team of paleontologists. And they were shocked when they thought, for giggles, that they would treat it like a bone and would do the equivalent of demineralizing it, removing the mineral, that's both the mineral that fossilizes it and also the mineral that's apparent in the bone. And what they got looked rather like a cooked bit of chicken. Which is interesting, the cartilage that's all kind of floppy at the end, which is interesting because when you run amino acid sequence analysis of T-Rex amino acid peptides, their closest match is actually chicken, which I find slightly comforting, actually. Anyway, so this 19-year-old pregnant T-Rex thigh bone presented an opportunity. And not only that, when they looked with electron microscopy at the materials, they were quite sure they were seeing cells, and they were seeing blood vessel fragments, and they were able to extract proteins and peptide sequences and run the peptide sequences. And of course, the community welcomed their results with open arms. No, they didn't. They said that's impossible. It's 68 million years old. There's no way that protein can persist more than 10 million years. And even that's a stretch. So they worked extremely hard to prove that they weren't lying, and the materials they found were not contaminations from either the experimenters or from bacteria. Bacteria do have collagen as well, but to say that bacteria collagen is like mammalian collagen is like saying I look like a pufferfish, which please don't say that. That's embarrassing. Also remember I told you the closest match with these peptide sequences that were taken from the T-Rex samples was chicken, not bacteria or not human. So they got some level of acceptance for their theories. In the meantime, collaborators, my closest and favorite collaborators actually, oh dear, it's recorded, never mind, I won't name them. Some of my longest collaborators suggested to me that we reach out to these people, and we did. And we said, hey, you love dinosaurs, we love dinosaurs. We also like collagen. Do you love collagen? And we all said yes, and we had a party. And then we took their amino acid sequences from the peptide sequences, and we mapped it onto my structure for the collagen microfibril. And as soon as I did this, this took about an hour or two to do. And when I finished this, I knew immediately that what we found was really significant. I didn't know why it was, I just knew it was. What we found is that these amino acid sequences cluster around sites that are critical for cell interaction and for extracellular matrix construction. They're not random. In fact, the significance score for the association of these peptide sequences with these amino acid sequences that are important for constructing tissue is well below 5%. It's the lowest p-score I've ever had. You have to look at the paper because I don't remember what it is. But it's outrageously low. So the correlation is obviously somewhat significant. And it took me a few weeks to figure out an explanation as to why this would be. And it's kind of compelling now that we think about it. Remember I told you the cell interaction site is extremely important. You don't want cells to get into the site until the tissue is ready. Well, it's here. And you have this cluster of sites, these peptide sequences, right around that interaction site. I hypothesize that this site is so important that collagen packing is designed to ensure that this site doesn't become available to the body until the body absolutely wants it to be. Therefore, it enables the same sequences and sequences near it to persist over long periods in geological time. There's absolutely nothing preserved on the outside of the fibril other than this little sequence here, which is buried beneath proteoglycan molecules anyway. And there's one or two things on the innermost layer. And there's less sites in the gap region than there are in the overlap region. There's also a couple other things that are going on for this. The amino acid sequences that are preserved over long periods of time are very low in acidity. So digestive enzymes tend to want to attack acidic sequences. So they're very low in acidity. It's from bone, and bone is naturally mineralized anyway, calcium and hydroxyapatite. And then lastly, it's fossilized. And mineral occlusion tends to preserve biological molecules much longer than they will if they're left out in the simple milieu. So you have these four compelling reasons, the first of which is biology does not want these sequences to become exposed until biology is ready for these sequences to be preserved. And as it transpires, this is a major, possible major reason why these sequences are also preserved for so long across geological time. And the last little thing that I'll tell you about is in our work to look at how tissues are organized and as it relates to disease and so on. We were working to characterize, this is from lamprey notochord, and this is from mammalian cartilage. And I told you the ingredients may be the same, but the recipe is different. The actual collagen fibrils here are identical. The actual fibrils themselves are identical. The organization is not identical. Same ingredients, different recipe. So we were looking at this for a variety of techniques that we've developed, and we accidentally took this and we made this. So we thought that these were whole collagen fibrils. What we accidentally did was make them into whole collagen fibrils, which were much, much smaller than the original ones. And then once we knew what to look for, we were able to do the same in mammalian cartilage. What did this was an antibody that we identified. And this particular antibody was able to molecularly dissect those collagen bundles more efficiently than concentrated guanidine hydrochloride or a Dremel. So the cartilage, 12,000 RPM. The antibody was more efficient at dissecting the tissue than the Dremel was. As it transpires, when we came to publish this article, the referees insisted that we retitle the paper to include the term plausible explanation for rheumatoid arthritis, plausible mechanism for rheumatoid arthritis. So we have an explanation for the early pathology, pathogenesis, I should say, for rheumatoid arthritis. And funnily enough, we have a patent in process that hopefully is on the last challenge to use this process to re-engineer tissues into possible cartilage replacement material as well. So at this point, I'll take your questions. Thank you for your time. Tom.
Just a very basic question on your last piece. You were saying that an explanation for rheumatoid arthritis is the attacking of the collagen by antibodies. Is that what?
In early pathogenesis of rheumatoid arthritis, there is a high incidence of anti-collagen antibodies and also proteoglycan antibodies. I hypothesize that early damage to the collagen fibril structure results in an antibody against those proteoglycans. Those proteoglycan antibodies tend to remove those proteoglycans. When you remove those proteoglycans, it's like taking off the compression straps on the log trolley, and they fall to bits. And that's why I use that image, is because that's what you're seeing in that slide that I was showing there as well. Then once you open up the collagen fibril like that, you suddenly have amino acid sequences that are in the milieu that weren't there before. And so the host's immunity picks up on new epitopes and starts producing antibodies against these new epitopes. And so in the end, your rheumatoid arthritis is a cascade syndrome, not just one particular autoimmunity, but a whole bunch of unique antibodies that are raised against itself. That just gets worse over time.
So your process would restructure the binding straps, or would stop the original antibody?
I don't think that we're going to be able to stop or reverse the rheumatoid arthritis that way. But what I know that we can do is use this process as an inspiration about how to re-engineer tissues to repair them. So osteoarthritis, we can come along and repair the tissue. And I'm absolutely sure of it. I'm sure that we would be able to not just repair the tissue, but restore it, and eventually develop knee joint replacements, restorations, I should say. And I'm bullish enough to claim that. The stopping the disease, the immunologically, is a different question. And I don't really have a great idea about how to do that yet, not because I don't have any ideas, but because I know it's very complicated. Yeah.
Just a quick question about a little bit earlier, when you were taking the X-ray photos of the collagen structure. Why specifically did you use X-ray for those structures?
Because we're looking at something that you can't analyze it from just the surface. I can't look at you and tell you how many vertebrae you have. Right? I could guess, but you may be about 0.5% of the population that have an extra one. So I would use X-rays and use a radiograph because they go through you. It's kind of analogous to that, the reason I'm using X-rays, I'm diffracting them. So instead of looking at the radiograph, looking at the contrast about which parts of your body absorb more or less X-rays, I'm looking at how the organization, the crystalline organization of your body, right, or more to the point, your molecules, alters the path of those X-rays. You had a question?
When the collagen models open up, is that then places that are exposed that go by one page more? No, well, obviously, but the actual stuff can't be treasured.
Yeah, the chocolatey center. Yes, and partly because it's these type 2 collagen fibrils. They're much smaller than type 1s, type 1 fibrils, so the radius of curvature is greater, which means there are more holes, and therefore it's just unbundling it will give access to most of the amino acid sequence. Whereas with a type 1 fibril, just a fibril bundle, unbundling it, you'll still see pretty much nothing until you start proteolizing it, which is probably part of the reason why you have rheumatoid arthritis being a type 2 disease rather than autoimmunity against type 1 collagen, which will kill you pretty quickly. You're welcome. Any other questions?
Just go with your title. When you showed your version of collagen and how it worked with that T-Rex sample, has that been, I noticed you had another...
There was a lot more on that slide, yes. Let's hope.
Basically, they try to do it again with some other...
Yes, so what I put on the slide, there was a lot more on there than there was in the manuscript. So the manuscript that was published last year, we covered just 2, the Brachioceros and the T-Rex. In the slide that I showed you, there were also pachyderm species that I included as well. There are reasons for that. We were as conservative as we could possibly be, not because we didn't think the pachyderms were interesting or good, but because this is kind of embarrassing to say. This is a paper that we hadn't worked on nearly as hard as a lot of other papers, and it's my single most downloaded and read paper ever, because it's dinosaurs. And it's almost awful to think about that in science there is a showmanship and political element in getting published, and you have to work hard and be absolutely sure to consciously hang on to your integrity whilst you make use of the wow factor. And that's what we were doing, is making use of the wow factor. In addition, though, we did have some very small concerns about the data analysis for the pachyderms. So I could have just hidden in that rationale, but I wanted to give you the honest approach, in full disclosure. Was there anything else you had on that question? That was it?
Basically, like, you said that they're still not quite believing that the legitimate thing. Have they tried with another process?
Nobody else is really going to try this, I'm sure. You know, it was a Tiger team that attacked this, and there's no other Tiger team that I know of that would be able to do that. That means like the dream team, the power wave in ice hockey or whatever. I think it'll just be a matter of time for people to finally get over their disbelief. So Sir Kelvin of the Royal Society at the turn of the 20th century said, this is as in the Kelvin scale, it is impossible for a man to travel faster than the speed of sound. Oops. We need not study any more physics. We have everything. We have Newton and his laws. Everything worth knowing in the universe has already been determined. Oops. And science tends to be pretty conservative. And the same thing that's good about scientific opinion and not taking any old wacky idea is also bad because it will not take on any wacky idea, which may in fact be innovative, or a brand new discovery that's wiped away years of misperception. The misperception about how long biological molecules will last is some fairly limited experiments a long time ago that have entered into the canon of scientific opinion, rather than being revisited how I was able to identify the MHC1 site on the collagen fibril. There are two ways on that. The antibody was raised against the sequence for the MHC site in collagen, and I worked out where the antibody bound. Secondly, there had been some amino acid sequence suggestions that it might be around there as well. The electron density analysis, that would be easier to take the computer aside and show you if you're interested later on. But it's essentially, you could look up the term multiple isomorphous replacement. And it's essentially, isomorphous replacement is a misnomer. It really is multiple isomorphous addition, but you'll find it under MIR, not MIA. And essentially, without altering the structure of the unit cell, so you have the unit cell being the same dimensions, the same shape, and mostly the same contents, you add material to each unit cell, or actually not even most. You need maybe about 5% of the overall unit cells to get a good signal. So a significant minority of the unit cells in your sample, you add another material to it. And this is actually how I solved the collagen structure in the first place, was using iodine and gold derivatives that were much, much smaller, however, than this, and used that for a phase trick to work out what the missing phases were from the structure factors. But I'm going to be using terms like that, which will make most of your people fall asleep. So I'd be happy to explain that to you more later, if you're interested. Any other questions?
Since you brought up like how dinosaurs are more related to chickens and everything like that, have they actually been using?
I didn't say that.
I'm saying it then.
Fair enough.
All right. So have they actually been using your work? Because I know they recently had me shipped at, not from the reptile amphibian, but to there. Have they actually been using your work for that too?
Gosh, I hope not, because it was an anecdotal observation on my part. I mean, to be fair, I remember growing up and hearing theorems being put out, or hypotheses, I should say, that a large number of dinosaurs were more likely became aves. Back 30 odd years ago, I was aware of that. So I don't think it would be my work at all.
Well, I'm just wondering if they've been using your work to basically give more credit to it.
To take my name in vain, I'm sure. But to be fair, the peptide sequencing, the major player on that is Mary Schwartzer, who's the paleontologist collaborator. Yes, I did some analysis of the sequences. It took me a few seconds to take the amino acid sequences and run them against the database in Switzerland and go, oh, look, closest match is chicken. So I'm kind of reluctant to take any credit for that, to be honest, even though it was me and the team who did work that out. But I would say it would be much fairer to say it is a general awakening and awareness that the birds and certain sections of the dinosaurs seem to be more similar than dinosaurs and mammals. Although a paleontologist would come along and shoot me in the leg for saying that, because I'm pretty sure there's a whole bunch of dinosaurs that are closer to mammals. To say that dinosaurs are more like reptiles or aves or mammals, I think is a little bit of a misperception of what we're classifying as dinosaurs. We could replace the term dinosaurs with monsters. And then monsters or dragons, right? Just to use what they used to be called long before we got into the 19th century when the fossils were first being dug up. It's a general classification. It's not a systematic classification of what those organisms are, as far as I know. So T-Rex, like chicken, yes. Brachiosaurus, can't remember. I can't remember if it was chicken or not. I certainly implied it. Any other questions?
Let's thank Dr. Orgel one more time.
Thank you.